think we transition immediately to the next presentation in this session, which will be given by Laura Garrison. Laura Garrison is a PhD student in the visualization group and at MMIV, and I'm really maybe it's back now. Great. And I just wanted to say that I'm really, really happy every day that we could win you to Bergen from across the Atlantic, because you are enriching our environment, not only in the sense that many of us do, bringing together the technical side and the medical side, but you as a medical illustrator, you even more enrich us with yet another perspective. And in the talk that you are going to give, I'm happy to mention you will comment on some recent research which this week was very positively evaluated at our best journal in the field. So we are all very happy and proud today. And thanks for explaining it to us now. Laura, please, the stage is yours.
Well, thank you, Helwig, for that introduction. I'll try to follow it up well then. So this was part of an ongoing collaborative project between here at the Viz Group and MMIV in Bergen and the Otto von Guericke University in Magdeburg and the Center for Behavioral Brain Sciences, also in Magdeburg. And the main focus that I'll have for today's talk is the application of a visual analysis tool that we developed called DIMLIFT on a clinical cohort dataset for cerebral small vessel disease, which was provided by our collaborators in Magdeburg from the before times when we could still travel, and hopefully we'll be able to do that again soon. And so we've talked about a lot of conditions that fall under the umbrella of cerebral small vessel disease. I'll call it just CSVD for short, because it's a bit of a mouthful. But it's a broad term for abnormalities in small blood vessels that are in the brain. So these can also be termed as white matter changes or white matter lesions. And so I've got just an image here showing degrees of severity from mild to severe. You can see those white matter changes in the brain there. And patients with this can be asymptomatic, but they can also have more serious presentations. So you can have balance and walking problems, dementia, cognitive decline, stroke, among others. So it's a relatively serious, possibly condition to be aware of. And it also has a fairly high prevalence in the aging population. So this study that we've talked about quite a bit in the last day or so, the Rotterdam study, there was a study born from that by De Leo. Yes, all right. I struggle with pronunciation sometimes. De Leo and colleagues looked at 1,100 subjects aged 60 to 90, and they found in this cohort 95% occurrence of these white matter lesions. So not only is it a prevalent condition, it's important to be able then to diagnose it and then follow up with good treatment for this. So it's not this very small disease that sort of hides out in the background. And so the problem here is that the etiology of CSVD is not well understood. So as, again, I think has been touched on by the proceedings in the conference, it's a hot topic in neurological research, partly because it's a broad classification of diseases, so it's difficult to really understand the factors that are important in diagnosing this, because you can have them come from lifestyle, from imaging data, from family history, and so forth. And this is an ideal scenario, we think, to then apply visual analysis and visualization to try to identify these biomarkers that are relevant in a very open and exploratory way. Visualization has proved quite useful to solve this task in the past, and we thought this would be an exciting way to apply this now. And so briefly now, talking about the characteristics of the data that we worked with. This was a largely tabular data set that we were working with. And it was characterized by being fairly wide and shallow. I mean, we're not talking thousands of dimensions, but it still is a fairly overwhelming amount to work with for a clinical researcher. So 193 dimensions for 307 patients. And as a terminology definition here, dimensions mean variable, or it can also mean a column in this data table. And we had 168 dimensions that described various demographic, laboratory, education, and lifestyle information. And an important detail here is that these were mixed data types. So we had quantitative data and categorical data, which presents an extra dimension of challenge to work with here. And we had 24 additional dimensions. So we didn't look at imaging data in the form of actual images. What we had were structural volumes that had measures extracted from them using a T1 weighted MR data. And then this is a problem that I think plagues almost all clinical cohort data sets. We had some missingness, 76% missing data. The reason for this can be from missed appointments. It can also be from, patient A doesn't need all the same battery of tests that patient B needs. So there's a whole gamut here, and that added a layer of complexity for us to consider in our approach. And so when you think about how this is normally analyzed, experts usually would use SPSS. For those not familiar with this, it's a fairly robust statistical analysis tool. It's usually easiest from our interviews with the clinicians and from doing a pairwise analysis with a couple of the clinicians, actually, that it's best when the hypotheses are already formed. Otherwise, you spend a lot of time reviewing the data, trying to find interesting subsets, and kind of cycling through over the process of many, many, many iterations. So it's a relatively lengthy process. For one of these pairwise analyses, it took 2 hours to find one kind of interesting subset. So it can be painful, especially if you're not already intimately familiar with the data that you're working with. And another test that clinicians would typically do in their analysis is perform some type of dimensionality reduction. And the reason for this is fairly simple. You have a ton of data set or a ton of dimensions, and it's really difficult to figure out what's going on, and so you want to reduce it to a smaller number of dimensions that still can explain the variation and explain that data set in a similar way. So this is this idea of going down to the intrinsic dimensionality. And they usually would do this with factor analysis. But even with this, so you get these dimensions reduced, but it still is possibly time-consuming to identify something that's interesting. And you still might lose the more subtle and interesting patterns that are present within your data by doing this dimensionality reduction approach. And so the challenge that we had with applying our visual analysis tool to these data were to see if we could give our users a means to organically explore and identify interesting patterns in these high-dimensional data sets. And this is something that we don't really envision as ever being a replacement for SPSS. It does an awesome job doing the thing that it does. But we wanted to be able to add some sort of precursor step. So if you didn't know the data super well, that you could find the dimensions that were interesting, that were maybe biomarkers, and then you could plug that into SPSS and just go for it. And so the core idea of our approach is these dimensional bundles that we're forming. So this is a standard approach with factor analysis of mixed data. And so the standard approach runs the factor analysis once, and you get the set of principal components, which are these super dimensions, where all 10 data dimensions are contained inside at varying contribution levels. In contrast, our approach does an iterative factor analysis of mixed data, and for each iteration, we look at the first principal component, and we extract out those dimensions that meet a defined contribution threshold. So those then form the basis of these dimensional bundles here. And it's a much sparser set of dimensions that we then run the factor analysis of mixed data on again. And that produces this structural hierarchy of the dimensional bundle that you see here. And so for each dimensional bundle, we keep the first and second principal component. The reason for that is the first principal component typically shows the most variation, second principal component the second most, and so forth. And we wanted to show a little bit of reference for the first principal component. So if you show like a scatter plot, then you can kind of see if there are extra clusters or any extra information formed within. And then the next layer is the set of the individual dimensions that contribute to that dimensional bundle. So what you end up with is a set of dimensions that contribute very similarly to the overall variation. So you have this possibility that maybe this group A is, these interesting lifestyle-related dimensions, and then B is body measurements or something like that, so height, weight, and then BMI or something like that. And then the last piece that we have here, so I said we do this iteration, But if you have dimensions that don't meet this contribution threshold that we've defined, we don't just throw those out. We keep those, but as unbundled, so then you can compare them directly with your bundles. And so then this is where we incorporate the user to allow for exploration and modification of the bundles themselves. And so this is looking at the CSVD data that our users provided to us. And so we keep each bundle as an axis in the parallel coordinates plot with glyphs that give information on the composition, as I've shown there, with information on their contribution to that dimensional bundle and the loading. And some interesting things already that the users found was this contribution of nicotine along with intracerebral hemorrhage in paravascular spaces. Another interesting finding was lacunes and microbleeds being grouped together. They said that this was not discoverable easily in SPSS. We allow this drilling down into a dimensional bundle to see clusters formed from the plots of the principal component scores against each other, which you can then flip to see how the contribution of the dimensions change between the first and second principal components. That's one way to detect interesting patterns of variation. And not only can you explore these bundles, you can also edit them. So if there's a dimension that you're interested in looking at within the context of a bundle, like APOE, this is a known biomarker, so the user was interested in seeing what was happening there. You can add that, it'll recalculate the factor analysis, and then you can look in and see the individual contributing dimensions and how they correlate within this bundle. And you can create subsets here along the score plot, or you can create them along the individual dimensions. And here, with the subset, you get a very quick view of the characteristics of this subset selection. And so this thing here I'm calling out because this is what took our analysts 2 hours to find, and we reproduced this in a few minutes. So this was exciting. And then just a few more points to bring up. This little bar here is present in all the individual dimensions and in the bundles themselves. And this is a completeness meter. So it tells you essentially how reliable your, in this case especially, how reliable your bundle is, because if you have a lot of missing data, you might not want to trust it as much. So we give some sort of indicator of that. And then we have also these disks along here. These are all white, and that means that these aren't contributing as meaningfully to the bundle as other dimensions that might be red or blue. And so to wrap up with a few take-home points from our collaboration with the clinical researchers in Magdeburg, through using DimLift for their CSVD data, they were able to openly explore the data space and have some new interesting findings, like this lacunes and microbleeds together grouping was exciting for them. I know, like Astrid mentioned this in her talk, so our study did not include cognitive data, and for something like this would be very helpful to have included, and this would be an interesting follow-up study to incorporate in the future. I mentioned the missing data problem. This is something we had to deal with somehow. Imputation is an interesting and also terrifying area of research to explore by itself. Our handling of this was fairly simple. We looked at 4 different methods of imputation and tried to see what worked best or just how much of an effect there was. And what was interesting was that we had the same number approximately of dimensional bundles produced for each of the imputation methods we tried. There's some variation within that, but we ultimately gave that control to the users to decide what imputation method they would like to go with. And then I've talked about this tool in the context of the CSVD data set, but it's made to be a fairly generally applicable tool. So in the paper that Helwig's mentioned that we submitted for the journal, we also tested this with a plant data set and a nutrient data set as well, with similarly interesting results. And with that, I'd like to thank you for your attention, and thanks to the conference organizers for inviting me to speak. I really appreciate it.
Thank you, Laura, for this very nice talk that I enjoyed very much. I must say I enjoyed also the collaboration on the research very much, so this is a double pleasure. We have questions to you, and I think we have also a bit of time to go through them, so maybe I can start with Hauke's question. He's wondering whether your stepwise creation of these dimensional bundles is in possible conflict with alternative approaches to creating a hierarchy, for example, that you take out a target variable that you want to explain, or anything else. Could you comment on that?
In terms of like k-means clustering as an alternative, for example, or? Yeah. So our approach, hopefully, so we have this initial algorithmic creation, but then when users can modify the bundles, they can add back in dimensions that they feel like were maybe missed or would be interesting to include. So by letting the user go back in and sort of decide if they felt like the algorithm didn't do what they wanted, then we sort of work around that problem.
I mean, if I may, I wrote down similar questions that are kind of close to what Hauke asked, so I just follow up now with my own. One way of alternatively creating a hierarchy would be to do this semantics-based. I mean, if you have, for example, results from cognitive tests, they may stem from individual tests, and maybe you find it meaningful to group them by their origin, not necessarily by their statistical correlation properties or whatever. Is that possible with your approach? I mean.
Kind of. The way we have it set up now, that even if you do create your own bundles, we still will do a statistical analysis on that bundle. But this option to sort of go in after the algorithm has done its thing will allow you to create those bundles that you want. And
I think we do have an option for manual integration and exclusion of dimensions in the bundles, right?
Exactly. Yeah. But we still have it set up to run the factor analysis on that after.
Yeah. No. I mean, I think then you want to see it probably after you have defined your bundles.
Too. Yeah.
Very good. So next, I have a very nice question by Morten, and I think the answer is also very nice. And it is, what happens if you transpose the matrix, the data matrix, before you start, and then you do an analysis on what were the items that became the dimensions and vice versa?
That'd be really interesting.
I think it's called the dual analysis framework.
Yeah. Sounds very familiar. That's our previous project.
I think, Morten, we should talk more about this. We have actually done this. It's not instantly trivial, because if you think about it, after transposition, the values are no longer from the same domain. So you don't have this coherency that the values are relatable to each other. So you have to in between, you have to have a characterization step, for example, by statistics, and then you make these values comparable, and then you can actually do it, and it's very interesting for exploration. Actually, the image that Astrid showed from our earlier work showed this, where we looked at many dimensions this way. So, good idea, Morten. And I can next ask a question that Eric asked here. Could you comment again on the glyphs you present on top of the principal on the parallel coordinates axis? What are they exactly showing, and how much learning was involved for the experts in order to be able to work with the application?
I glossed over that one. So there's 3 glyphs. I don't think it's easy for me to go back since it was part of a video, but, or maybe actually, I've got it here. Yeah. So this first one here is, so we're using positional encoding, so the amount of fill indicates some sort of goodness of fit. So the first glyph here is indicating the eigenvalue. The second glyph in the middle, and so these 2 bigger glyphs are only present on the dimensional bundles because they're only relevant for those. The middle glyph is the completeness of the data. So the more empty that looks, the more caution you need to have when you're thinking about the results of your grouping. And then this glyph here is divided into segments based on the % contribution of each dimension contained within that bundle. And then we use a diverging color, so red and blue, to indicate the sign of the loading.
Very good. I think we take one more question from Bernhard Breim, who also applauds your talk and says that is a great talk and a really strong contribution. I must read this out. I mean, this is such a nice comment. His question is how well the physicians could interpret the results of the PCA.
So actually quite well, and I think part of it was the physicians that we worked with are also experienced statisticians, so they were familiar with going through dimensionality reduction, and they knew PCA and factor analysis, so we got lucky there. One of the clinicians that we worked with, though, was less advanced with statistical analysis, and it took him a little bit more time to understand. I had to explain to him what PCA was and roughly how it worked, but then he was able to still find interesting conclusions from this just by the way the bundles were made and the way we'd set it up. So that was really heartening to see.
Very, very good. I think we take all other questions offline. You will be around for a few more hours, I guess, here also.
Yes.
So let's thank Laura again for her talk.